3 days have been with you. 3 दिन हो गया ना? How has this been so far? We still have. This is a penultimate day. Ultimate day is coming tomorrow. I hope the course is also ultimate. So maybe we can have any inputs for you from your side about the course, because I am meeting you for the first time and I was thinking we should have had a ice-breaking session also, because I don't know some of you. So some of you seem to know biophysics. Am I right? Maybe quick introduction करेंगे. I should know each one of you. Yes, I probably know him, but still. Keep sitting, you know.
Just completing my PhD.
Completed. Oh, nice. टॉपिक एक टॉपिक बताएंगे ना क्या है?
Sir, my topic was design in vivo in silico designing and the synthesis and validation of novel chemicals to ameliorate Alzheimer's disease.
बैठे रह सकते हैं ना? You can keep sitting. पुनीत कौर. टॉपिक बता सकते हैं, मेरे को थोड़ा रिलेट करने में इजी होगा. If there is something I am going to tell, then I can relate better. Neuropathic. Which model? Liquid biopsy. Euro? Viral. Yeah. So it seems like there is a dress code from Central University Punjab. Black. Yes, right. Okay. थोड़ा सा ऊंचा बोलना है ना. Topic.
It's in the founding stages, but I'm planning to work on neurobiology.
Keep sitting and then introduce yourself. No?
Good morning, sir. My name is Varsha Bharadwaj. I am from Naipur, Nagar, and I am under professor.
Naipur?
Yeah.
Dr. Sharma's lab, no?
Yes, sir. I am currently working on Alzheimer's disease. Good morning, sir. I am Anvi Pandey and I am from Naipur. I am also working under Dr. Sharma, and my research project is on the standard disease.
You will have to introduce for everybody. Yes.
So my name is Riya Chanda. I am in my last year of masters in the biochemistry.
So she will be working on something related with brain, but she doesn't know as of now. Yes.
My name is and I am part of the biochemistry and doing my master's.
My name is Nisha.
ऊंचा बोलना.
My name is Nisha.
Topic of your PhD, you can tell. Huh? So you can also introduce yourself. So I am using this opportunity to know you all. That's also part of this course, that we should be knowing you. Maybe we'll come back to you. Yes.
I'm Chaitanya. I'm from BITS, Hyderabad.
BITS, Hyderabad. Wow.
So we are working on organelle interaction, ER mitochondrial peroxisome interaction during chronic ER stress in age.
ER and peroxisome interaction. Good to hear about peroxisomes after a long gap. ER and peroxisome.
ER mitochondrial peroxisome.
Very nice.
Sir, myself is Vikram and I am from Dean's University, pursuing my master's second year.
Jaipur?
Yes, sir. Under the coordination of Dr.
Khatri.
Khatri, sir.
Oh, Khatri.
And my topic is related to neuroinflammation in Parkinson's.
Great. So I didn't know Dr. Khatri student is also here. Great.
Sir, my name is Abhishek. I have done my master's in stem cell nutrition engineering from Punjab University, and my dissertation topic was effect of preconditioned secretome on depression model.
So maybe the last two can also introduce, and then I'll start formally. Louder.
My name is Nitya. I'm working with Professor What I'm working on is vegetative walls and low associated manifestations in neuroscience. So we are trying to understand stress associated manifestations and developing a therapeutic targeting ENS and combating CNS symptoms through it. That's where it's coming from.
So Ramya, you also go ahead. All right. So I'm one of the course coordinator with Dr. Tanzir, you know that, and I've been in Punjab University for last maybe 26 years now, and probably we'll be completing 26 years in another couple of days on the campus. So I've been trained at PGI. I've been pursuing an interest in neuroscience. I'm also a student of neuroscience only. I think this, I said, we still don't know much about neuroscience. All right, I picked up a topic which I don't know why I picked up, but I think it's useful. That's what I will say. So maybe a year back when the proposal was signed, maybe we had a different thought. Now the thoughts also keep evolving. So I'm going to talk about flow cytometry and how it can be applied in neurobiology. So I've put few applications of neurobiology, but all the applications that I'm going to discuss will actually apply to neurobiology also. So how many of you have used flow cytometry in your experiments? One, good. One hand goes up. I think, yes, two. So maybe few more can use it by the time we close this discussion. I think it is a handy tool to use for neuroscience also. There are few things that where it provides extra strength. People in, I would say, cell culture would use in and out flow cytometry, but using tissues also you can do lot of things using techniques like flow cytometry. So just to keep everybody long, the word flow cytometry comes from flow is fluidics. Fluid is very important component of this. Maybe in theory you read it in earlier classes also. Cyto means you're looking at cells, and metry means measurement. So this is something that we should be understanding. This is about a bit of history. Cells we saw for the first time, Leeuwenhoek. Then first apparatus for detecting bacteria using this kind of mechanics was in 1947. So they were trying to look at bacteria using the flow kind of mechanism. But the flow cytometers evolved around 1965 for using it for cells from tissues. And what came there was you could also add sorting into capabilities into these flow cytometers also. There were different kinds of evolution, but I would say this is a time when Hansberg from Stanford University developed a cell sorter that separated cells stained with fluorescent antibodies, and this term fluorescent activated cell sorter was introduced. So there are two terms that I will mention, flow cytometry and FACS. Both mean different things. Flow cytometry is I am just looking at measuring cells or some parameter of cells when it is moving through the flow. When I am saying fluorescent activated cell sorter, that means the cells are labeled. So I can then use it for detection and then I can also use it for sorting also. So there are two differences in the two terms that I am talking of. So variety of measurements can be made on cells, cell organelles can also be studied, and you can actually study any object that is suspended in aqueous environment. Does not mean always it has to be cells, and you will all agree by the time we close this. So this is already we talked. So there is a fluid, there is a cell passing through the fluid stream, illuminated light path is there, and then there would be some kind of instrumentation which we will learn little later. So it helps us to detect cell surface markers, intracellular molecules, factors, secreted factors can also be looked at, DNA content can be seen, few applications I have just listed here. Quantifying of cells can be done, I can know the absolute number, different properties of the cells. You can categorize cells and quantify them. So there could be mixed population, I can categorize within the analysis and then quantify them also. Subpopulation of the cells can also be looked at. Applications in all fields, neuroscience is not listed in this because that's a very specific domain. Immunology, hematology, pathology, microbiology, genetics, functional studies can be done. Cell culture studies we already said, toxicity studies, drug discovery, chemical engineering can be using this, biotechnology can be using this, agronomy can be using it, animal sciences, plant sciences can also use it. Alright, looking at the particle size. We talked about flow characteristics using different sizes. Bacteria was the first one we said. Look at the sizes, 0.5 micrometers. But when you go to eukaryotic cells, the size is bigger. RBC 6 micrometers, monocytes would be 14 micrometers. So these are the kind of particles that can be analyzed in flow cytometry. So one difference you see is their sizes are different here. This is the fundamental slide. This is the whole gist of my lecture today. So what you have is cell. When the light source falls on this cell, we have a possibility that would be forward scatter, there would be side scatter. So forward scatter will depend on size of the particle. Bigger the size, this forward scatter would vary. And what would be the factor on which side scatter would depend? Granularity of the cell. What kind of granularity of the cell is there would be side scattering. So I can use these two parameters only to separate population of cells. I am not doing anything but using these inherent properties of the cells to analyze cells. I don't have to do anything. Different cells, if I take blood cells, they will vary in size. We can go back to that slide. They will vary in granularity. We can separate them. Alright, if I do this, forward side scatter, I make a plot, I can get some dots, events. These are events are being picked up, the dots are indicating, these are plots that indicate events. So I can see some populations in these events also. One is a concentrated one at the bottom, one is a little more diffuse, there is another population I can see in the middle of the two also. So based on these two only, I can separate some cell types. When I am saying forward scatters, when I am saying side scatters, there has to be some detectors to pick up these signals. There will be side scatter sensors, there would be forward scatter sensors, and there would be laser beam, which will be the right source. Alright, we said all this, what we have been talking. Forward scatter is related to surface area or size we were saying, detected along the axis of the incident light. Side scatter, reflected as well as refracted light both are counted. related to cell granularity and complexity of the cell, detected at 90 degree of the laser beam. So both detectors are there. So this is one illustration of this. No, I'm moving on further now. What is this picture showing you? This picture, let's look at the graph only. It is picking up fluorescent signals. Forget about side and forward scatter. What you've done is you've labeled certain molecules on the top of the cell. So there are two kinds of labels that you see in this picture, or you can see two kinds of circles at the bottom, which I can say are the circles which are representing cells. One cell population is having less expression of antibody on the top or marker on the top. The second population is having more of that. So when I separate them, based on that, they will also show size variation is also there, and there is another variable that I brought in is fluorescent signal that I can pick. So based on this, I can actually analyze n number of parameters. What would I require if I have to analyze n number of parameters? So I said, using these, what have we done so far? We had forward and side scatter. Now we have added, maybe in this picture I could say one antibody, or we can say two antibodies. How many parameters I am looking at now? 2 or 3 at the most, maybe 4. How many parameters can we analyze in a flow cytometer? Anybody who will make a guess? At one time. Any guesses on number of parameters I can analyze? Guess, wild guesses are also okay, but I would say instead of wild guesses, intelligent guesses are better. How many parameters? It will just depend on antibodies. How many different kinds of, and the laser beams. So the capability of the instrument improves if I have more number of light sources. I may be giving you one example of a fluorescent signal using, let's say, one filter. If I add 4 filters into the flow cytometer, I am increasing my parameters to maybe 16 or 20 parameters, or even more. So it depends on the number of filters I have in the instrument. The cost of the instrument also goes up once we talk about number of parameters you can have, detect more filters means more parameters. But the only problem would be when I pick up my labels, I have to see they should not be, we'll talk about those, they should not interfere with the signals. But the capability increases moment I add fluorescent. So when I was only talking about forward and side scatter, I was limited to the parameters. So this is just a data from forward and side scatter. I have been able to pick up dead cells, bigger cells, size variation, apoptotic cells, live cells. Another example, blood cells, granulocytes, monocytes, lymphocytes could be picked up. Size, granularity variations. So there are few limitations of, so what I should not have talked actually about fluorescence in between, but my slide came in between, so I just mentioned that. So what are limitations with light scattering? Some information can be obtained. If I have two cell types which do not differ in size and granularity, I have a challenge. FSC correlates with cell size. FSC can distinguish living from dead cells. Side correlates with internal complexity, we said this. To distinguish between two cells size, we need both these parameters to be different. I said this already, that if they are not different, we have a problem then. If these two parameters are same, then no distinction can be made. This is a limitation of forward and side scattering. This is a plot of size and granularity from the alveolar fluid, from biopsy mice, so lot of things. But I will have to sort and pick up the population that I am interested, and we'll come down to that again. So then we set I can pick up other parameters to analyze. It can be based on cell surface markers, which I am labeled it as cellular antigens. Intracellular cytokines could be used. Enzymes could be used. Structural differences, that means some adhesion molecules. Metabolic differences in the cells could also be picked up. I can use it for analyzing metabolic changes. I can use flow cytometry for even knowing something happening in the mitochondrial environment. I can use it for study of intracellular calcium levels. A lot of things within metabolism can be looked at. And it says probably I could be just giving a tentative kind of list. 160 different parameters could be analyzed using this technique in different populations. We talked about this now. Adding fluorescent signals, you will have to sometimes fix a cell, incubate with an antibody, permeabilize that also, label, and then signal has to be acquired using whatever we said. So what is most important here? Two more important things come: fluorochromes and antibodies. So we know fluorochromes, these are the common ones. FITC you might have used in other techniques also. Phycoerythrin, PERCP, APC, many others. They could be small organic compounds. Then these have to be complexed with antibodies. This is what is a flow cytometry antibodies. So we all know fluorescence spectra. I don't know how to say. Has to be excitation wavelength, where this is what we are saying of the light sources, and there has to be emission spectra also. And I'll just skip this slide. We all probably know this. I will not talk about this. This is a picture I am revisiting that. We had explained this. You are picking up signals, fluorescent signals, instead of the other parameters that we talk of. I said you can analyze n number of parameters, but this n number of parameters would depend on this: emissions and excitations. We will want them to be apart so that I can detect those. There should not be any leaching, there should not be any quenching. These are the things that we need to be careful when I'm picking up, selecting my labels. So there is a lot about this. Probably I will not talk about this. Is just picking up the emission spectra of these. So I will not prefer APC and PERCP together. Maybe I will use them which have emissions far apart. So when I am talking about emissions, look at now the complexity of the machine also improves. So I will have light sources of particular wavelengths. I will also have different detectors now. So machine becomes more complex. So what is the most important thing in a flow cytometer? What do you think would be the most important thing in a flow cytometer? Heart of the machine.
So what is its specificity, like how efficiently it can produce?
I'm saying heart of the machine.
Like,
What are you saying is not incorrect. I'm saying heart of the machine. Where you think is the whole emphasis on the machine?
the light source.
I was thinking something different, bit different. Fluidics is the most important actually, first. Then comes the optical system. So light sources and the detectors would be the second most important. Fluidics is the most important, and that's where also the problem lies also when you run the flow cytometry. Yes.
Sir, 160 parameters in total or in one run?
So I would say the one run parameters, usually people have gone to something like 16 or 20. Could be a little more in theoretical sense, or maybe some people have tried that. But I would say more than 6, 8, 10 people don't go, because you don't have those probes that efficient which will be segregating that. But people have gone to 16, 18 parameters at one go, using different light sources. Yes.
Sir, and the parameter which we can analyze it will depend upon the laser source we are following.
Definitely. So I can have UV, red, green, blue, whatever sources I use will add to the capability of the machine. So when I am buying a machine also, I decide then which light sources I have to use. Depends on how much I have in my wallet. I can increase the capability if I have more money.
Sir, can you go for FITC, PE and BBC?
This or earlier one? Earlier? So what I am saying here is, this is just telling you about excitation spectra of these. So excitation for each one is at a different wavelength. If I say FITC is getting excited at 488 nanometers, if I go to APC, something like 600. That means probably here it will be green filter, here it could be red filter. I could be wrong in telling you the filters. Maybe I'll show you a table which has the filters then. So based on this, my excitations vary and my emissions will vary based on what probe I am picking up. So most common one is FITC. PE is another common one that people use. So you will get antibodies that are already labeled with these from commercial sources. So you can have primary antibodies labeled with this. You can have secondary antibodies labeled with this. Both are available. So if I have to use same secondary body, same probe, I can use secondary bodies that are something like ELISA also. But ELISA doesn't give you primary antibodies already labeled with a fluorescent signal. So you will use secondary there. But here you can have primary antibodies also available. So that means one step. So most of the times we'll do one step. We are not doing any secondary antibody into this. But possibility of that is also there. So I said this is a typical flow cytometer. Now you can see forward is being seen, side is being seen, fluorescence 1 for FITC detector, FL2 for PE, and then there is FL3 for other two dyes also. So I can also have more number of detectors depending upon what signal I am using. So I have already talked about this. Maybe we will see this slide again. You can go to BD Biosciences website. It tells you what dyes I can use, what are the emissions, what are they, and it will even guide you which of these will not interfere with other signal also. This is another pictorial representation of the same kind of things. Blue-green laser. These are the dyes that can be used. Violet laser can be used. Then you have a capability of using Pacific blue, M cyan. Red laser, you have a capability of this. This is what is adding to the number of parameters you can analyze. I have not added one more filter into this. That is UV source can also be added into this. That's also useful, but it becomes more expensive also. So this is now dyes that earlier we were talking about molecules or capabilities wherein we had antibodies labeled for detection. Now we are saying molecules themselves can also be used. We don't need to go for immuno labeling of cells. Acridine orange, RNA DNA binds, discriminates between them, lysosome labeling, amino actinomycin D, DAPI for nucleus chain, you will need UV here. That's where it is adding a capability here. Propadium iodide, you can use with red laser and a few other. So likewise, I will not highlight on all. Hext is common one people use, but you need a UV source for that. So this is where you will have to decide the stains. So if I am using DAPI and propadium iodide, so if I have to use, I don't have a UV, I probably cannot use two of these filters. I probably will have to go with propadium iodide then, or likewise. Reticulocyte labeling can be done with another dye. So look like the capabilities increase. This list can go on further. Maybe I'll just pick up one or two here, not all. It is giving you a fluorescent list of fluorochromes and it is telling you what is the advantage of each. For example, Lexaflor, it says substitute for FITC, better stability and intensity. So based on these kind of parameters, you will decide which one is what I want to use. Rhodamine green, let's say, I will say again the same kind of properties. Maybe we will pick up something PerCP, long red dye. So if I have to go to red range, I probably would use this. So I'm just giving you an idea, but then you can, once you dive down into it, you will find better answers to what I'm sensitizing you with today. We can also have specialty dyes. The earlier list we did not have specialty things. I was actually looking at giving a thought that probably something is missing in my slide, but this is what I wanted to talk. If I have to do calcium measurements, I can do these two dyes. If I have to look at mitochondrial membrane potential, JC1 is also used, which is there in the bottom list. There are others in this list also, reactive oxygen intermediates people might be using or could have used also in the past. And cellular membrane potential things, other things. Okay. For Golgi apparatus, we have something different. So tandem, you can use tandem fluoro probes for the reason some of these fluorochromes would impact the other. So you will have to find which one work together. That will be tandem fluorophores, and these are some of the examples of tandem fluorophores. I am not getting into this as of now, but this is a complete list. This is from BD website. BDs are market leaders. I am not marketing for BD, but broader information from there. So you can see the kind of lasers, you can see the excitation lasers, you can see the fluorescent channels can be used, and the fluoroprobes that BD recommends for that. So this actually is a serious component of analyzing parameters in a flow cytometry. If you attend a week-long workshop on flow cytometry, this is what they are going to teach, mainly how to decide the fluoroprobes or how to analyze the data. Running is not that difficult part. So my idea is to sensitize you, tell you in brief. Maybe some of you can use it. It's not a difficult technique. And maybe I'll tell you some applications which are not in my list also today. How can I present my data? I can present my data like this. What this data shows is number of cells and the events, simple graph. So it tells you me two populations, one which has low CD count, other has high CD count, and the one which has low CD count has more events. Likewise, I can now analyze two parameters. When I am analyzing two parameters, I can have CD8 versus CD4. So my plot can come out like this, and I can say at least there are two major populations which can be differentiated here, this and this. So there are different ways of presenting this data. Histogram, one we saw earlier, 2D plots, the one which showed was dots. We can have 3D plot, cell quest is the name that is given. I can have 3D plot attractors as another way we can do that. This is one of the instruments, Fax Caliber. We have one in our department called LSR Fortessa, which has 3 lasers I guess, and probably we bought it maybe 10-15 years back. So system has this fluidics, it has optics, it has electronics. This is majorly the components. I will not spend too much time on this, but I will just go through. I said heart of the machine is fluidics. Why fluidics is important? I want to answer from you. I did not emphasize why it is important. I forgot to mention one thing very, very important. So what the role of fluidics, we said that fluidics role is to have a system that there is one cell passing through, or you are able to analyze one cell at a given time. That's the whole idea. If there are cells that adhere to each other, probably the purpose will be lost. So there has to be certain parameters, and that is why we are saying air filter, that means airflow is also there. Sheath fluid is the fluid in which you are carrying the cells, and they will pass through a narrow capillary, which is where you are analyzing the sample. And whatever is then passing through is then collected through the waste. But if I want to improvise the capability, I can not let the waste go. I probably can use them as separation facility. This is what is fluidics actually now. So sample is shown in a reverse order. You can think that the picture is in the other order. You have a sample, but when it passes through this capillary, they are all in as single cell. So this is the most important part. That's why I said heart of the machine is the fluidics. And you spend money, the recurring cost is also on the sheath fluid in which the cells move in as a stream also. So excitation lasers and rest of the things are all part of it. All right. So I can vary the pressure You can have a machine with low differential pressure, you can have high differential pressure. The rate at which the cells move will be more in the case of high differential pressure, but in each of these cases there has to be single suspension. In this case you only can see one cell, you can see two cells, but advantage of that would be I probably would be able to capture more events. So optics we all know what will be there, detectors and all. We don't want to get into the instrumentation business because that's something I don't want to spend time, and this is how the optics looks like, and that would be so. And there is a lot of emphasis on optics because look at the kind of detectors that you are using, look at the kind of sources of light you are using. So the optics also has to be very, very important, and that's why the machines are expensive. This we already talked. Maybe this is the architecture of one of the instruments that we can talk. So you can see all these filters that are picking up the signals. FL1, side scatter, FL2, 4, I showed you in the early picture 3, and likewise you have excitation and forward and side scatters. Measurements, we talked about these. This is just an example of picking up cells what is the cell being picked up? CD14 negative, CD18 plus. That means antibody would be binding, these red ones are the antibodies that are binding to CD80 plus. CD14 is green. So based on these I can separate CD14 versus CD80 cell types. So already we will discuss this figure. I can have 4 different populations based on these fluorescent signals. This is the dot plot we were talking. So this is CD14 versus CD80. So I have seen cells which are negative for both, they will all come in this population. CD40 positives are coming here, CD14 positives are coming here. The ones that pick up both could be in a broader population that is somewhere here. Histograms could be there for these same data. This is just CD14 in the left and signal being picked up here. I think there is a problem in my picture. I should have had the other picture, same picture got pasted here anyway. So one more thing in flow cytometry is gating. This is one picture, forget about what my circles say for a minute. What I can do is now, this is my population coming from forward and side scatter. I can further analyze these populations, this population and this population by gating these, which is a software based application. So I am gating these and further analyzing these populations because this might have subpopulations within itself. So that is the advantage of having both gating. So gating also becomes a very, very important component in flow cytometry. So you have seen R2 has been separated into populations. Likewise, R1 has been separated into different populations. You can do gating and separate them into different populations and then analyze them making different kinds of plots. So what all applications can come now? Phenotype analysis, intracellular analysis, DNA analysis, cell function analysis. One thing I have not talked is cytokine bead arrays. How many of you have heard of this? No. So I said you don't have to analyze cells, you can analyze any kind of particles. And that will be what is called cytokine bead arrays. That, as the name suggests, I can analyze cytokines. How do you normally analyze cytokines? How do you normally analyze cytokines? कैसे देखते हैं cytokines हम? Yes. ELISA, basically you do ELISA. So if I do multiple cytokines in a given sample, this technique will be useful. Little amount of sample, I can analyze 10 parameters, 20 parameters. That's the advantage. We'll come back to this and explain that. Cost is a little higher because you're using more antibodies also in the same sample. So you can't expect cost would be down, but sample saving is the most important here. Intrinsic, we talked about this. Extrinsic, when we are adding agents to analyze it, it will be called as extrinsic parameters that can be analyzed. So we talked about all these. This is kind of still a repetition. This also we have already seen. different kinds of population, phenotypic analysis, we've talked about these. I can analyze lymphocyte phenotyping can be done. This is very useful in immunology. Yes. Which one?
Can you comment on how cell's intrinsic fluorescence would impact the extrinsic fluorescence? Suppose if the cell has its own intrinsic fluorescence.
You're saying the intrinsic fluorescence, some of the cells might have their intrinsic fluorescence, but that signal will be very weak. That will be very, very weak. You don't expect that to be there because you are using labels which are much stronger and exciting its specific wavelength. So I don't expect that to be part of it. Yes, there could be quenching, but those kind of issues can be sorted out. Maybe we can have a detailed discussion on that. If I have my slides, I will show you that. But otherwise, quenching is also important because some signals can even mask your signals also from the cell itself. Or each label can also mask each other. What you are saying is the inherent fluorescence, but eukaryotic cells you don't have it. You will have that problem mainly in plant cells because you have more fluorescent pigments into plant cells. So you don't have it in eukaryotic cells, that bigger challenge. Minor will all be taken as baseline. So not an issue to worry about in animal cells. So I can do different populations, all these populations. I don't have to tell you that, you have understood that. I can then look at I have been able to analyze so many different parameters from, so immunology labs in a hospital will be using this. All these big hospitals, immunology labs will have flow cytometers to analyze these kind of parameters, the populations of cells, because the treatment depends on some of these. Fine, allergic reactions, you want to analyze the cell population, just not antibodies. Intracellular analysis, I talked about these. When it comes to intracellular analysis, I will have to do two things: fix the cells, permeabilize the cells. Same kind of techniques that I do with histology are used here also. Histology or immunohistochemistry. We use a detergent, could be tritone, could be anything else. We will also use formaldehyde, 4%, for fixing the cells here also. So in the tube you are fixing the cells, you are permeabilizing within the tube. This is a cartoon to illustrate that. The membrane, now the antibodies are entering and you can pick up the signals from within the cell. So easier is always cell surface. You can do it within the cell also. So a lot of things, cytokines I said could be detected intracellular also. We might have an interest to know how much is the production of cytokine. So there you will need permeabilization agents. This is an example to that, tell you that. So I can stimulate the cells to produce some cytokine, analyze that, and it says formaldehyde, glutaraldehyde could be used, permeabilization, and then you can do staining and then pick up different kinds of signals. All right, I will skip some slides in the interest of time. I can also analyze signal transduction parameters, many of them. All right. Intracellular staining in activated lyse blood cells. This is talking about intracellular stains. That means I am picking up what is the signal being picked up. If anybody can read the slide, my slide. It says intracellular signal. STAT3 is a transcription factor. You are using a phosphotyrosine label STAT3 antibody to pick up how much is STAT3 expression inside the cell. So I'm just giving an illustration. You don't expect me to go deep down into it because that will take a lot of time for us to pick up what this each figure interprets. But I just want to give you an idea that I can pick up intracellular molecules by permeabilization and fixation, and in this particular slide it has picked up STAT3 signal using a phosphotyrosine label for that particular antibody. This is most important. DNA analysis can be done. Neurobiology will have application. Cancer would have more applications because cycle is more important in there. So same ethanol detergent, you could have nucleic acid dyes. These could be the kind of dyes we could use. You all familiar with this dye. When I do the analysis, I can pick up cells in different stages. G1 has the maximum DNA content, followed by S, G2 or M phase. So this could be the picture that I can put it in other words, and I can analyze different cell populations. Within this entire population, I can pick up sub G1 stage cells from that population by using this gating. So I have a population, within population I am screening a subset of populations. All right, we talked about some of these, and I'll come back to apoptosis, which is again where it's commonly used. Cell proliferation, BRDU stain is used, propadium iodase is used, calcium we already talked. Intercellular pH determination can be done by SNRF1, oxidative metabolism, TCF, DA, membrane potential, we talked some examples. How many of us are familiar with annexin 5 stain? May not be. In apoptosis, membrane asymmetry changes. So what you see is, during apoptosis, what you've seen is the red dots have moved to outside. So now they can bind to annexin 5 FITC conjugate, and that can be a new signal that I have emerged now. Annexin FITC stain can pick up these apoptotic cells. Adds to my strength in analyzing these parameters. Look at what we have got now: live cells, necrotic cells, and apoptotic cells. We have been able to analyze cell death using some stains. This is FITC stain, annexin 5 stain. Using PE, some populations I'll not get it, but I just wanted to give you an idea. You can also look at mitochondrial membrane potential using JC1 stain. So you will pick up signals on what is the membrane potential. If it's changing, JC1 stain would vary, and that also would be picked up. Proliferation assay, I said BRDU stain can be picked up. This is where I wanted to spend time with you for a few minutes. Now, another technique which is powerful and handy is cytometric bead arrays. I said cytokine bead arrays, but it should have been cytometric bead arrays, because mainly we are using for cytokines. So you have a bead which has antibodies. So antibodies are now capturing their antigen. So this is the antigen, which could be cytokine, picked up by this bead. What happens in, so how I am analyzing multiple parameters? This is the answer. You are using beads of different sizes. Size differences would be picked up. Different fluorescence intensities, different colors of beads could be there. You are adding capabilities. I could analyze 8 different parameters for cytokines using this. Multiple analysis simultaneously, reduced sample volume requirements, reduced hands-on time, wide dynamic range of fluorescence detection, no problems in terms of interference. This is a data from cytokine bead arrays. So many cytokines analyzed. This is another way to plot that same data. Anyway, you can also make standard curves using this, not getting into that. You can have, this is another data from cytometric bead arrays. Open configuration. I can decide what should go on my beads. Not only the pre-made ones. I can have 30 different parameters, and I can use clustering based on red and NIR fluorescence intensity and dual lasers and things like that. Anyway, we've talked about this. I don't have to say much. We can improvise this technique a little more. All right. This is what would be another capability in flow cytometry. I don't throw the stuff that comes out, separate that into different populations based on voltage. The ones which have no voltage are in blue, or no response to the voltage, are going straight. Others will be deflected based on the charges. You can do the sorting. People use it, the cost adds to the machine. So if I'm buying it for 1 crore, it will maybe go 1.5 or 1.78 crore. So you will spend more money. But why would you need a cyto sorter? Because if you want to use the cells again for doing something, then you need a sorter. Or your purpose is to sort the cell, then you will be using it. Otherwise, if my purpose is only analysis, why should I spend money on sorter? All right, can I make the technique even more simple? Yes. Magnetic sorting. I can have magnetic beads labeled with antibodies and do the sorting. There are a lot of techniques available where you can use magnetic sorting. Magnetic sorting could be a step before flow cytometry also. More cells in a population, I want to enrich the population, I'll use magnetic sorting. In this particular case, it's saying sperm sorting is being done, but I can use it for any kind of cell. I only need antibodies that or something that my population can bind to a magnetic field. So what it says is magnet, it holds that population, I demagnetize the environment, that same population is left out. So I can separate cells using this magnetic, and these are less expensive also. This is where I'll have another dimension added to my flow cytometry experiments. Now coming to the real topic that I had to touch upon, but all that I told you can also be used for otherwise also in neurobiology, but I will repeat so that we have an emphasis. There is something that I will add extra. I said you can analyze any kind of parameter. You can analyze nanoparticles also using flow cytometry if I can pick up the differences there. If I have two populations, I can use, I'm making nanoparticles, I'm loading with something, I know there are inherent differences in the population, I can use flow cytometry, any particle. Exosomes can also be analyzed using flow cytometry. Just giving you another example, we've used that some time back, and this is the capability. Cell population analysis, we can do that. Phenotype and marker analysis, we talked about that. Intracellular state, we talked about that. Cell sorting also we talked about that. In brain that becomes important. Why? Because we have different population, neurons, microglia, astrocytes, few other populations. There magnetic sorting will also be helpful. I'll also come down to one more technique. Receptor quantification is cell surface marker. I want to analyze the number of cell markers on the receptor on the cells, specific cell types within the hippocampus, striatum, and see their expression. I can do Western blot also, I can use this also. Advantage with this would be, if I'm separating the cell population, I'll know which cell type is contributing to this. Neuro-oncology, yes, I can pick up glioblastoma cells and then analyze them in terms of cell cycle, in terms of other markers which will be specific to cancer and things like that. So two more examples I will talk to you is, one is this. So I said you can dissociate tissues, remove myelin and cell debris, separate one set of population, let's say CD11 bead incubation, magnetic separation. Which kind of cells will be separated within CD11B? Which cells are we separating with this? CD11B is marker for easy है, मुश्किल नहीं होना चाहिए. Microglia. So microglial marker. So you should be able to collect CD11B positive cells. Negative also I can pick because I know where the negative have gone. That will be in the first part of the sample. I can use even simple technique if I don't have a magnetic separation technique available. Look at what my right side says. Dentate gyrus have been isolated. I've just picked up from the net. So why dentate gyrus should not be asked because it can be any section of the brain. Tissue can be dissociated. Homogenization. Then you can separate them through cell strainers. They are available at different porosities. I can separate cells and then analyze those cells. And it says you can label those cells then with antibody and then the rest of the things. This is, I find, is more interesting even. How many of you have known synaptosomes? If I homogenize a cell, my neurons pinch off and form vesicles like this. So these become functional units coming from neuron. I cannot analyze neurons. I can analyze synaptosomes as a functional point of neurons. This is just an example. where they have gated the synaptoneurosome population. And the same population has been analyzed using electron microscopy also. That what I said you, the synaptosomes, these are the circular particles that you can see are synaptosomes. So I can analyze these also using, and I can do any kind of study using synaptosomes because these are also vesicles now. They are kind of smaller cells which I can analyze in terms of calcium influx. I can do oxidative stress measurements. I can do any kind of parameters. I can look at the presynaptic terminal markers on this also. At the same time, I can look at these populations using microscopy also. So this is a summary of what I said: cell phenotyping, cell cycle analysis, cell functional microbial sorting. We didn't touch on this. We can add few more things to improvise on the data, and it can be high throughput, purity, single cell sorting, multiplexing. All those things would be beneficial. This is all the summary. All right, I'll just say it is a valuable technique for studying cell diversity, cell signaling, changes in neuronal cells during disease, can even sort and isolate specific cells for further study. So I just wanted to give you a window to this. Maybe rest of the things you will read a little more and then maybe try to have this in your research if possible. Thank you. Any questions? Most welcome.